We're moving on from this cyclization chemistry, but I want to just reiterate a few things before we do so. Remember, when we cyclize sugars, we're going to do this kind of attack that I have shown here, and it can happen in one of two ways. When the carbonyl is pointed down, you're going to end up with an alcohol pointing down. When the carbonyl is up, you're going to end up with the alcohol pointing up. That's the difference between the beta, or rather the alpha and the beta anomer. Now I also want to say something that I haven't said before, and that is the cyclization chemistry happens all on its own, and it's totally reversible. And I want to show you what that means on the next page. So let's go on to that next page, and this video is called Mutorotation and Reducing Sugars. And the first concept we want to talk about is the mutorotation idea. Sometimes I write this down as spontaneous anomerization. Anomerization, kind of hard to say. What I mean by that is it's the spontaneous conversion between alpha and beta forms of a sugar. Let's pause for a second and have you sort out which of these two sugars is the alpha and which is the beta form of glucose. That'll be a good quick review of what we did in the previous videos. Remember, that skill is going to start with looking at the anomeric carbon, which in this case is carbon number 1. It's the carbon that's attached to two oxygens directly. And then we're going to look at the anomeric alcohol and compare it to the high-numbered exocyclic group. We see both of those groups on the same side, that is above on the left-hand structure, that is beta, whereas they're on the opposite side of this structure that makes alpha. Now the key here is that if you were to drop either of these structures into water, they would spontaneously convert back and forth from one to the other. And in order to understand the chemistry of that, you have to understand that it would go through the linear form. So when I draw linear glucose, I draw it out like this with an aldehyde in the carbon at the top, an alcohol at the bottom, and then I do right, left, right, right. So let me say this again. If we start with beta, it can uncyclize to become the linear Fischer structure, and then recyclize to become the alpha, and then uncyclize to become the linear and back to beta. So this is a complete equilibrium, and as it turns out, it's about 64% in the beta. It's about maybe 35% roughly in the alpha, and about 1% in the Fischer structure. That tells you something. That is, the cyclic Haworth structures are much more stable and therefore more prevalent in the body than the Fischer structure. However, you cannot interconvert between the two cyclic structures without going through the Fischer. That's why we need to know all three of these structures and how to deal with them. So this is a spontaneous process. It's going to happen if you just throw this in water. It'll equilibrate out to these ratios. However, it is spontaneous only if the anomeric alcohol is what we call uncapped. What do I mean by that? Well, I mean if it's a free alcohol. That's the only thing we've seen so far is an OH here and an OH here. So these can mutorotate because they can uncyclize and they are uncapped. There's nothing attached here. We're going to see going forward that we can attach things to these alcohols, other sugars, phosphates, nitrogens, things like that. And when you have anything but a free OH, these things are stuck in the form that they're at. If you have beta with something attached here, it cannot uncyclize, and therefore it can't undergo mutorotation. Okay, that's a skill that we're going to practice, but I want to move on to something called reducing sugars. This is something that shows up later in our class, and it shows up in the lab, and it's kind of old chemistry and old terminology that we just feel like we need to cover. and it's called reducing sugars. And it has to do with something called the tolerance test. It's an old qualitative thing. And the way the tolerance test works is you mix a sugar with some silver ion, and if it has a free aldehyde in it, it'll reduce the silver and plate it out like a mirror. And you can see this in this little picture that I threw out there. It can plate out silver on the bottom of a test tube. Maybe you've seen this done in chemistry class. So, like I said, this is a test that is for the presence of a free aldehyde. So let's note our linear structure has a free aldehyde, and it would form a positive tolerance test. These cyclic sugars are not having free aldehydes, so they are technically negative. However, remember we said that the cyclic sugars can spontaneously form the linear sugars, and therefore if we put these cyclic sugars into water, you're going to see that they're actually going to turn out positive because they are going to become linear, they're going to have a free aldehyde, and they're going to therefore be able to make that silver into silver plate. So let's finish this off by saying reducing sugars are those that have an aldehyde or can form one spontaneously. Like we said above, the middle structure has a free aldehyde, that's positive. The ones on the outside don't have a free aldehyde, but they can form one spontaneously, so all of those are reducing sugars. Keep in mind, we're going to see things that have caps on them. Remember, if we put a cap on here or here, it cannot come over and become a free aldehyde, and those would be negative. And we'll see examples of that moving forward.